Thank you. My name is Yong-Sun Bahn. And first of all, I thank Joe for organizing this wonderful meeting and then giving me a chance to have a wonderful sabbatical in your lab. And I also thank all the Hyman lab members for supporting me during my sabbatical time. So I'm going to share so I'm glad to be here and present my recent work. I've been doing this project like for 7 years, and then finally we have some kind of getting results. So I'm going to share this result to you. I don't want to spend too much time about the Cryptococcus neoformans or fungal pathogens because I assume that many of you are expert in this field. So the long-term goal in my lab is kind of to understand the complex signaling network regulating growth and differentiation and stress responses and virulence factors and host interactions of the fungal pathogen. And by using this information, we try to find a novel antifungal drug target. So for this purpose, we utilizing Cryptococcus neoformans as a model system because Cryptococcus has a very good genetic tools because it's haploid cell, so it's much easier to knock off the gene compared to Candida albicans having a diploid strain, and also has a wonderful animal model system. So if you look at some, like if I simplified some signaling network in the one of the signaling pathway, so when you have some environmental signal, most of the signaling pathway composed of the sensor, like a receptor like a protein, and the signals are transmitted to the multiple kinase or sometimes the phosphatases like a calcium pathway. And eventually this signal goes to the multiple transcription factor, which is subsequently regulated some downstream target gene, which counteract or respond to the signal. And then depending on the signal, there could be multiple signaling pathway could be there. And to make this story more complex, actually, a lot of times these signaling pathway, they crosstalk with each other. Eventually you're going to have this kind of very complicated signaling pathway, which like systems biologists like to use. And if you look at this complex signaling pathway in Cryptococcus neoformans, and then we found some very, probably many of you probably know that. So when you compare the kinases in the Cryptococcus genome versus the transcription factor, as you can see, this is a blast matrix kind of presentation. The blue means it's highly conserved, the conservation, and then the white means it's the less evolutionarily conserved. And then Cryptococcus looks like they're about like 185 kinase, and the Cryptococcus have like around 180 transcription factors there. As you can see that they have a highly conserved transcription kinases, however, and the transcription factor is more evolutionarily divergent. So which means that this evolutionarily divergent transcription factor could be regulated by the evolutionarily conserved kinases. So one of the example I can show you that a couple of years ago we published the papers and then about the function of the unfolded protein response pathway. And this UPR pathway is basically the sensing the misfolded or unfolded protein accumulated inside the ER stress, inside the ER organelles. And then what happened is this UPR pathway consists of the very evolutionarily conserved the ID1 kinases. So ID1 kinase has both kinase and endoribonuclease activity. And if you compare the Cryptococcus ID1 or Saccharomyces or human ID1, they're very evolutionarily conserved. However, the downstream target transcription factor working downstream of ID1, such as like HEC1 in Saccharomyces disease or XBP1 in human, if you just blast search by using those amino acid sequence information, you cannot find any kind of ortholog in Cryptococcus neoformans. So we did a lot of screening to find out what could be the downstream target of the ID1, and then we found the HEX1, which is another type of the BG transcription factor. However, the sequence is quite different, and also when this HEX1 cannot complement Saccharomyces HEC1 mutant, and actually if you try to express the HEX1 in Saccharomyces becomes sick. And then actually we nicely showed that this ID1, HEX1 is a bona fide downstream transcription factor of ID1, and then both of them are required for the virulence. So this is a good example that you have very evolutionarily conserved kinase, but you have very evolution divergent transcription factors in Cryptococcus neoformans. So my lab, we thought about it. We could just probably, what if we kind of analyze this kind of relationship in the more genome scale? So first, the research goal we had it a couple of years ago, and let's decide, like, identify the all of the Cryptococcus transcription factor, and then let's disrupt the TF mutant, construct the TF mutant library, and then through analyzing phenotypic trait in vivo and in vitro, and then we could probably can have some more comprehensive Cryptococcus transcription factor networks in there. So we started this project a couple of years ago, and then we selected 178 putative transcription factor, which has been predicted by the DBD database. This is publicly available in the DNA binding domain specific database. So this database actually specifically predict the sequence-specific DNA transcription factor, not so excluding any kind of general transcription factor or sequence non-specific transcription factor is excluded. And then after that, for this 178 transcription factor, we designed this gene disruption cassette by either split marker or by using conventional overlay PCR method. And then we transformed the Cryptococcus strain. So we used the H99 platform strain having normal virulence by the ballistics transformation. And then eventually, after we performed the first screening by the diagnostic PCR, and then we confirm all of the mutant by the southern blot analysis. So, and also we try to have the high quality TF mutant library. So we try to get at least 2 independent mutants for each genes. So finally, we made it 2 independent strains for the 155 transcription factor genes. So we got total 322 strains out of it. So the other 23 genes, we couldn't knock it out. For example, some of the transcription factors were found to be essential, such as like a HSF1 was found to be essential, and there are several essential genes there. So and then after that, we decide to analyze the mutant by testing different in vitro phenotypic trait, and also we did some larger scale virulence assays by using those mutants I'm going to talk about later. And then this is the list of the phenotypic trait we measure. We tested the growth at different temperatures. We monitored the mating and virulence factor productions, and we tested the stress responses of many different conditions. And also we tested antifungal drug susceptibility too. So since this data, we have 30 different phenotypic traits. Also, each one has a different concentration, and then for total 322 strains. So, which means that we have more than 20,000 data points there. So, if we simplify this database, so we got this kind of TF database like that. So, if you look at this database more carefully, if you can see that this is the transcription factor, and then when you have some ortholog in the Saccharomyces genes, and then we put that name here, and then this is all each in vitro phenotypic trait. The red means is that highly defective or highly sensitive phenotype, and the blue means is highly resistant or enhanced phenotype. And then also we put all the RNA-seq data too. So the red means is highly expressed genes in the 6 different conditions, and blue means a very low expression kind of thing. So if you want to see the real data, and I told you there's more than 20,000 data points, so we cannot put all this data in the paper. So we decide to generate some publicly available TF phenome database. So this is if you go this database, and if you put your favorite gene numbers here, CNH number, which is the serotype A, H99 gene names, ID, and if you put the information here, and then you can retrieve all the genetic information. And also you can get some RNA sequence data out of it, and then amino acid or protein sequence. And then you can see how we knock out this gene. And this is what kind of primer we use, and then this is southern blot data is here. Then we also put all the in vitro data in this website. So it kind of it's better than just having the supplemental data. So if you go to this transcription factor database, you can get all the data out of it. So since this is the antifungal drug summit, so I don't want to talk about all the other phenotypic traits I discovered. I mainly try to focus on what kind of virulence-regulated TFs we identify. Okay, so mostly the pathogenicity is determined by 2 factors. One is the virulence and infectivity. Okay, so in the classical way, you can do the virulence assay by just mouse survival test, like you saw this kind of data through this summit. So as you can see that if you have a wild type of mutant, you can just infect the mice, 10 mice per group with the wild type and mutant, and then you can monitor the survival curve like this to measure the virulence. If you usually, you also have a complement strain, and then you get additional groups like that. And then also you can measure the infectivity by after you infect the mice, and then after a certain time point, and then you recover the infected organs, such as the lungs and brains or kidney. And then you can calculate the CFU per gram tissues, and then you can say that the wild type, the mutant infected mice as a lower fungal unit, which means that you have a low infectivity, something like that. So this is the classical fungal pathogenicity test. However, you cannot do this kind of classical fungal pathogenicity test when you have like 322 strains in total, because you have to order like 4,000 mice, which is almost impossible to do it. So we need some kind of high throughput in vivo virulence or infectivity test. So for the high throughput in vivo virulence test, we decided to use this surrogate host model, such as the Galleria mellonella, the wax moth model. I didn't invent this model because this model has been in the community for a while. And then it turns out that this is quite, this Galleria mellonella model has been very proved to be well correlated to the mouse virulence data. And one of the beautiful part of this is that it's very cheap, it's very convenient maintenance, it's very easy to inject. It takes like 3 seconds to inject the strain into using this pull leg. You see that there's a multiple legs here. So you inject the Cryptococcus through these pull legs there. And then also you can monitor the virulence factor at 30 degree and 37 degree, which means that many cases the pathogen, when you have some thermosensitivity problem, then you lose the virulence, right? And then if you want to know that non-thermosensitivity kind of related phenotype, then you can use this Galleria systems also. So by using this Galleria system, so we did the Galleria virulence study for the first screening for 155 TF mutant, 15 Galleria per group. So we isolated 63 TF mutant in the first screening. And then we performed the second screening for this 63. So we tested second independent mutant for each TF gene from the first screening. And finally, we isolated TF17 transcription factor mutant were confirmed to show reduced virulence at a statistically significant level. So this is the list of the genes here. The yellow marked genes that has been already reported or in some mouse model, something like that, so which means it's highly correlated to the mouse data. And then some of that, the novel genes, you show that like a 2 independent strain show the similar reduced virulence patterns like that, as you can see like that. And then also we performed some larger scale infectivity tests also by using the signature tag mutagenesis based mouse model. So in these cases, when we create the TF mutants, so we put the unique signature tag in the marker. So which means that by using this, each TF mutant, even though they have the same selection marker, so each TF mutant, so we decided to use the 50 different signature tag. So each TF mutant, 50 TF mutant have a different set of the signature tag. And then what you can do is that by having the signature tags, so you pull the strain together, then you infect a single mice, and then you can compare the input versus output by using this is not QRT-PCR, just quantitative PCR method. And then if the certain TF is the virulence factor, that if that TF gene is deleted, then they may not survive well in the output. So you can compare that this data and that data, and that is we call the STM score. So we did this experiment for all 322 strains, and then you can get this kind of data, which means that some of the strain cannot survive very well, and then mostly they're like a wild type strain. And as a control, we use the STEL 50 and ID1 as a control. This is a virulent control. This is a virulent control strain because we already reported previously, and this one before, so we used this one as a control strain. So we got this kind of data, and then combining all this, and then we got the final several like 30 genes we identified through this. And then this group of genes actually have been also found in the Galleria model, which means that these 10 genes have been found in both Galleria system and then mutant-based STM analysis, which means that it's highly likely to be involved in virulence of the Cryptococcus neoformans. If you look at this data more carefully, and then also, you didn't see that, it's kind of weird. So, and then when we compare our TF virulence data, and then also the Candida albicans, like the Sandy Johnson group, and did some like systemic analysis of the Candida albicans virulence transcription factor, and then when we compare our data and their data, and then this is like some of the 5 genes commonly identified, which means that these transcription factors could be the very broad spectrum antifungal drug target. And then one of them is the NRG1 and NRG101, is the ND groups that have identified this transcription factor. And then also CRG1 is a calcineurin transcription factor, have been also found in our data and the Candida albicans data. And the USB101 and ZEP104 are kind of, a little bit new. And then if you look at the phenome data of this, and then as you can see, JIP104 has a reduced capsule productions like that, so that might be the responsible for the reduced virulence. However, in the case of the USB101, they has a higher actually capsule production. However, in terms of the melanin production, USB101 is highly defective in the melanin production. So that might be the reason why the USB101 and JIP104 has a very low virulence in that model. However, some of the virulence-regulating transcription factor doesn't have any phenotype. For example, PDR802, this one has really highly reduced STM scores like that, and also in the highly reduced in the Galleria model. However, if you see this phenome data, they don't have any phenotypic trait whatsoever. So we have to figure it out what this PDR802 and for some other transcription factor is really doing in the Cryptococcus neoformans. And actually, interestingly, some of these virulence-regulating transcription factors are also involved in the mating, which is responsible for the production of infectious propagules like this. So they completely block the mating, cell fusion. They cannot produce the proper pheromone. So these genes are also kind of mating regulators too. So when we try to correlate the in vitro data with the virulence data, as you can see that the virulence, in many cases, the temperatures or a lot of environmental stress is kind of highly correlated into the virulence. As you can see, the mating is kind of less correlated to the virulence data. And finally, I want to briefly talk about it. So as I told you that the question is, and then how this evolutionary divergent set of the transcription factor could be regulated by the conserved kinases. So we kind of did we employed the same approach for the kinases also. Because it's the kinases, is compared to the transcription factor, kinases are more favorable drug target, as you can see. So we also did the kind of same things for the kinase network too. So eventually we knock it out. I mean, we found out the kinase is much more difficult to knock it out than transcription factors. So in these cases, we originally identified in the Cryptococcus genome, so we found that there's about 185 curative kinase in Cryptococcus neoformans. And then finally we kind of made a success in making a kinase mutant for about 117 right now. So this is ongoing process. But anyway, we got 2 independent mutant strains for more than 100 kinases. And then right now, so we finished we did a lot of some virulence study by using Galleria for this mutant. We are planning to do some STM analysis for this kinase mutant library. But if I show you some of the Galleria data for these 105 kinase mutants, so when we test the Galleria virulence study for 105 kinase for 2 independent mutant strains, and then we identify the 26 kinases actually exhibit reduced virulence, the statistically significant level. If I categorize these genes, and we found a lot of known kinase genes, as you can see, the cell wall integrity pathway, MPK1 pathway genes, or Hog pathway, MEK kinase, or cyclic MP pathway, and some UPR pathways I talk about it, RAM pathway kinase, and SNP1 or glycogen synthase kinase have been identified. And also a lot of some genes involved in growth of cell cycle have been identified, or nutrient metabolism. These are arginine, methionine, cysteine uptake is involved in methionine metabolism. and URK is the pyramid and nucleotide metabolism. And the signaling pathway is GAL83 is the beta subunit of the SNP1, and IPK1 is inositol. I'm going to talk about pentahispoid later. But anyway, it's also there's a tRNA modification or retrograde trafficking. Also, there's some unknown kinase we identified it. So if you look at the blast matrix, as you can see that most of the kinases are highly conserved. Then we kind of interested that some of the kinases like this. For example, this kinase is only conserved in the Cryptococcus or some Basidia and some plant. However, they're not have like a strong similar ortholog in humans or other Ascomycetes. So that could be, I think, the very good Cryptococcus specific kind of target things. And also, so there's some other genes that have some low homology in the human ortholog. Now, if you look at what is this gene, it's actually inositol pentakis phosphate, so IPK1. So if you delete the IPK1, you have a very reduced virulence like that. It's actually amazing. This mutant has like phenotypes, so many phenotypes. It could completely block the mating. They don't produce melanin. They don't produce urease. It's completely sensitive to high temperatures, even though they produce a large amount of capsule. But if you look at the stress sensitivity, it's kind of sensitive to almost everything. So this IPK1 could be the really good anti-cryptococcal target. So right now we made the cDNA, and then in collaboration with the Sang-Yi Lee's lab in the Seoul National University, he kind of made a success in the made of crystal structures of IPK1. So kind of then now we're working on some to make some the IPK1 specific inhibitors in the near futures. And another transcription, another kinase is interested in the GAL83. As I told you, this one also is the human ortholog has a low similarity to the GAL83, and the GAL83 also has a highly reduced virulence. If you look at it, also it's kind of very sensitive to multiple stresses, especially hydrogen peroxide and the cell membrane stress, especially kind of very synergistic with the amphotericin B also. This one is kind of interesting. Like this is a known kinase we call the virulence-regulating kinase, and then even though they're like if you delete the VRK1, then you have a highly increased, little increased capsule production. However, even though they don't have like obvious phenotypes, so although they show some sensitivity to some fludeoxinol, which is the fungicidal drugs, but we have to figure it out the function of this more carefully. And also this CDC7, which is a cell kind of involved in cell cycle control, and also is important for the virulence. And the last things I'm kind of BPS15 is kind of interesting target because a lot of times that that is known that like usually the Golgi to endosome export kind of trafficking system is important for the virulence because it's involved in capsule production or other things. But it's not very well known that what about the role of the retro trafficking from endosome to Golgi in the virulence of the Cryptococcus neoformans. So we found that one of the BPS15 is actually involved in this retrograde trafficking from endosome to Golgi. So if you knock it out BPS15, they have a highly reduced virulence in Cryptococcus neoformans. It's actually the reason why I'm focusing on this is that also BPS15 is known to be required for the Candida albicans too. So that could be the very broad antifungal drug target. As you can see, published by the Scofields groups, and as you can see, if you delete the BPS15, you have enlarged the vacuole morphology like this, and then you have a highly reduced fungal burden in the mouse study. And then we found the same kind of thing in the Cryptococcus neoformans. So if you delete the BPS15, you have a highly enlarged vacuole morphology compared to the wild type. And then what happens is if you delete the BPS15, and then it makes has some growth defect, but that growth defect is more severe at the high temperature. And also, as you can see, they're sensitive to many of the stresses, especially very sensitive to cell wall and ER stresses. And actually, in Candida albicans, it is known that if you delete the BPS15, that cell wall integrity, such as CRG1 or calcineurin pathway, is upregulated. So might be same things happening in the Cryptococcus neoformans too. So to wrap up my talk, so right now, so what we're going to do is that we kind of systematically analyze most of the kinases and transcription factors. Right now, we try to make some connection between kinase or between kinase and transcription factor. We're doing a lot of phosphoproteomics analysis to find the direct target of each kinases. And also, in future, so we're going to do some transcriptomics or chip-seq analysis to find some downstream target of the important transcription factor. And then having this data, then we could have more clear pictures about the complex signaling pathway in Cryptococcus neoformans. All this work is done by the, I will tell you that like, I think so about 30 to 40% of these mutants have been made by undergraduate students. So when they spend 2 months or 3 months in my lab, they make 1 or 2 years. And then, and this, I have 4 graduate students in my lab, so he just graduated, and then all the previous semesters students. And then the Eunji Jung's kind of helped us to do some STM analysis, and Guillaume Zangbong shared the RNA-seq data with us, and then Yong-Wan Lee made the beautiful TF mutant database. Also, he's working on making the kinase database too. And the John Hyman provided all these beautiful Galleria systems for us. And then the Anna and Taeyups here, and they help us to do the Galleria study. I'm going to stop here and then taking some questions from the audience.